Hey, good morning or afternoon. Today we're going to talk about some of the main methods used in modern biotechnology and how these techniques are applied in the fields such as medicine, agriculture, and forensic science. In this presentation, we're going to focus on four important methods, and they've all been developed since the 1950s. And we'll start with a quick overview before presenting each one in more detail. So let's get our pen going here. The first one, DNA sequencing. Determining the order of the bases of the DNA, which is A, T, C, and G. And if you don't remember what those stand for, it's adenine, thymine, cytosine, and guanine. DNA sequencing is important because it's used to study how DNA works and to screen for genetic diseases like sickle cell anemia. Recombinant DNA technology allows you to take a gene from one organism and transplant it into a different organism. One of the main applications of rDNA is to turn bacteria into factories for producing useful molecules. For example, the human gene for growth hormone has been put into bacteria so that they produce human growth hormone known as HGH. It is this HGH that some athletes have used to enhance their performance despite health risks and get themselves fined and kicked out of sports in the process. Somatic cell nuclear transfer is a method for replacing the nucleus of one cell with a nucleus from another. Since the nucleus contains all the DNA, this will produce an identical copy of the first cell, so it is a cloning method. This is used in animal cloning, such as Dolly the sheep, the first cloned mammal. And then finally here, the final method is polymerase chain reaction, or PCR. It's like a copy machine for DNA. Starting with just a few molecules of DNA, PCR can make millions of copies. It's important because sequencing of other techniques usually require a large amount of DNA to work with, so this does a good job of copying it. One application in forensic science where it will allow investigators to recover even a tiny amount of DNA from a crime scene. Now let's look in more detail here. For sequencing DNA means that we're determining the order. There are many different methods and a lot of research being done to create newer and faster techniques. Sequencing DNA is estimated to be a $5 billion business each year, so if one company can develop a better way to sequence it, it's going to make a lot of money. One approach is to attach a tag, each different chemical label, to each of the four nucleotide bases. Fluorescent dye molecules make very good tags, as they are different colors and can be detected automatically using a laser and a special camera. Several kinds of automated sequencing machines use this approach to read the order of the different tags, which gives you the order of the bases. So as you can see, each one of these is a different color, so this is a different tag. Whoops. So this is a different tag for each one. So that might be A, that might be G, etc. Or actually it says this is G, C, 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 T, so it tells you the order that it goes in. Several kinds of automated sequencing machines use this approach to read the order for the different tags. This is just one way to sequence DNA, and there are other methods. Talk a little bit more about the Human Genome Project. It sequenced one complete set of human DNA. A complete set of an organism's DNA is known as its genome. HGP, or the Human Genome Project, was led by the U.S. government, which provided most of the funding. The project ran from 1990 to 2003, so it took them 13 years to get this done. A human genome, the DNA in one complete set of 23 human chromosomes, contained about 3 million bases. Remember, those bases are A, T, G, and C. Having a complete genome sequence from a human being provides a map that makes further research easier. For example, if a scientist isolates a segment of DNA from a human cell, he or she can tell very quickly where in the genome it came from. This helps decipher the connection between DNA and cell function. Different people do have somewhat different DNA sequences, but the differences are relatively small. Usually only about 1 out of every 300 bases is different between people. So even though the HGP is just one complete set from a mix of individuals, it can still be used as a map for everyone. Only about 10% of the human genome is composed of genes, with the other 90% having other functions such as regulating what gene is active. Now, DNA sequencing has gotten cheaper and faster. It took about 13 years for the first one, like I said, and it cost about $3 billion. But the HGP also drove many advances in sequencing technology. We can now sequence DNA much faster and cheaper. Companies are starting to offer whole genome sequencing to individuals. In 2015, you can have your entire genome sequenced in about 10 weeks for around $7,000. It is so much cheaper to sequence human genomes now because of the original genome can be used as a reference, so you know which one is supposed to go where, more or less for every single person. Now that one lab can sequence billions of DNA every week, we must rely on sophisticated computer programs to store it all. And the computer analysis of DNA sequences that is part of a bioinformatics, a growing field that combines computer technology and biology. So this is a new growing field of science that you can get involved in. Now we're moving to that second one, recombinant DNA. Genes from one organism are inserted into the genome of another. Now consider people who need insulin in order to treat diabetes. The old way of producing insulin was to harvest it from a slaughtered cattle. But that was expensive and difficult, and some people were allergic to cow insulin. So biotechnology researchers isolated the human gene for making insulin. Then they used a special enzyme called a restricted enzyme to cut open the circular genome of E. coli bacteria. Most bacterial genomes are circular. Next, they used a different enzyme to insert the human gene for making making insulin into the bacterial genome. This created a new form of life, an E. coli bacteria with a human insulin gene. Because the DNA code is universal, the gene works the same in bacteria as it does in DNA. For humans, so the new recombinant E. coli bacteria produced human insulin and do all their offsprings. In 1982, human insulin produced this way became the first drug made by using rDNA to be approved for use in humans. So it's only been around since 1982. It was the first drug developed by a biotechnology company, Genentech, which was sold in 2009 for about $50 billion. Sales of recombinant human insulin amount to about $1 billion a year. It's kind of crazy. It's also got some ethical concerns, though. So we can transfer genes between species and create new life forms. This raises some obvious ethical and safety questions. Scientists held a special conference in 1975, soon after the first rDNA experiment. At the conference, they drew up initial lists of guidelines for safe and ethical rDNA research that they all agreed to follow. For example, they agreed not to carry out rDNA work on highly pathogenic or disease-causing organisms. Later, different government agencies formalized these rules and added others. But there is still concern that rogue scientists, or those working in countries without rDNA regulations, could accidentally or intentionally create dangerous organisms. There are many kinds of bacteria that cause serious diseases in people, but most can be treated with antibiotics. But there are genes which make bacteria immune to certain antibiotics. What if someone used rDNA to make a disease-causing bacteria immune to antibiotics? The disease would go crazy. Next, we're talking about the somatic cell nuclear transfer. Is a method for removing the nucleus from an adult cell and transplanting it into another individual's unfertilized egg cell that has had the nucleus removed. So you're replacing one nucleus with another. The egg cell with the implanted nucleus grows and divides. Since the nucleus contains the genome, the result will be a clone, an individual genetically identical to the donor. The cloned cells can be implanted in the uterus of the surrogate mother if the goal is to produce a cloned animal. This was done in mammals for the first time with a sheep named Dolly that was produced in 1996. So again, they took the nucleus out of one adult cell, put it in the egg of a cell that had the nucleus taken out, and then inserted that into the uterus where it started to grow like a normal baby. And so this is how we've had clones. The polymerase chain reaction was developed in the 80s as a method for making copies of DNA. And to analyze or sequence DNA, you typically need a substantial amount of it if you're going to just look at it over and over. So this was a method to provide that. If you only have a few molecules, you could really do anything with it. But using PCR, you can make millions of copies, which you could then analyze in different ways in sequencing it. PCR is a three-step process. First, the starting molecules of DNA, known as the template, are heated in order to separate the two strands of DNA. Remember, they're stuck together like a double helix we learned last time. Second, a pair of shorter segments of DNA called primer binds to the template DNA. The primers mark the segment of DNA to be copied. Such copying is also known as amplification. And finally, next, an enzyme, usually Taq, fills in the section between primers with new nucleotide bases. Now we have two copies of each template DNA molecule. Then the three-step PCR cycle is repeated, giving us 4 copies. Since the number of copies will double with each cycle, 10 cycles will produce 1024. copies, and 20 cycles get you over 1,000,000 copies. PCR doesn't copy an entire molecule, just a segment between the primers, which usually cannot be more than about 5,000 bases long. Do you remember how long the whole thing was? Researchers design the primers to target the segment of the DNA that they are interested in, and PCR is fast, taking just a few hours, and relatively inexpensive. It is one of the most common techniques in biotechnology and is used in almost every application that involves DNA. So, how does biotechnology help detect and treat diseases? It's revolutionizing the world. Sequencing DNA allows doctors to determine if a patient has one of thousands of genetic diseases known in humans. Once a genetic disease is detected, treatment can often be improved in the patient's health. So they look and they match their DNA versus what we know genetic disease DNA to look like. Cancer tumors are typically caused by mutations to the DNA. By sequencing the genome of a tumor cell, doctors can better predict which anti-cancer drug, which is called chemotherapy, is most likely to destroy the tumor. The general approach of using a patient's DNA sequence to choose the best drugs to treat their condition is called pharmacogenetics, or more broadly, personalized medicine. It is currently a very popular area of research. Many drugs are produced using rDNA technology as well, like human insulin and human growth hormone. It is estimated that over 250 such rDNA-produced treatments are currently available, and more are approved every year. Finally, cloning and stem cell technologies, such as those made possible with SCNT, are being investigated for the potential use in medicine. Research is aimed at cloning a patient's cell in order to grow replacement tissues that would not be rejected by his or her body. This kind of work is still experimental, but it's very promising and the focus of much research. There are many other ways that our biotechnology is used in medicine, so this is not a complete list. Scientists and farmers use biotechnology to help improve agriculture. Over 13 million farmers around the world use agricultural biotechnology. rDNA is used to produce varieties of crops that are resistant to disease or pests. For example, corn borers are insects that ruin several $1,000,000 worth of corn each year in Nebraska alone. A soil bacterium can naturally produce a compound that kills corn borers but does not seem to harm most other insects or animals. The genes that produce the compound were isolated, and the rDNA technology is used to insert those genes into corn. The new strain of corn, called BT corn, is resistant to the corn borer. Crops also have been enhanced using biotechnology to supply more nutrients. For example, vitamin A deficiency kills over 1,000,000 people each year, so scientists use rDNA to add a provitamin A gene to the rice. This golden rice is being tested at a few farms, but it has both supporters and opponents. Food contamination with disease-causing bacteria is a problem. As anyone who has ever had food poisoning knows, the Center for Disease Control estimates that 1 out of every 6 people in the U.S. gets a foodborne illness each year. Of those, over 100,000 are hospitalized and about 3,000 die each year. PCR and DNA sequencing can detect which species of bacteria have contaminated a food item. Scientists can use that information to trace the contamination back to its source, which is why we get food recalls on certain things. Biotechnology is also increasingly used for environmental applications. Some organisms have been genetically engineered to detect pollution. For example, the rDNA glowfish were created to detect water pollution by glowing in the presence of certain compounds. Biotechnology may also help to clean up pollution. For example, researchers have used rDNA to engineer oil-eating bacteria. These have not yet been used on a broad scale. Endangered species have usually lost much of their genetic diversity. This can create many problems, such as inbreeding. Effective conservation plans require understanding the genetic diversity, and PCR can be used to retrieve DNA from droppings so as to not disturb the animals. Then DNA sequencing can provide important data on how much genetic diversity is left. Finally, biotechnology is harnessing plant and animal products for fuel. These biofuels may be effective, sustainable substitutes to fossil fuels like gasoline. Biodiesel is made from a plant or animal fat, is available in many cities, and some vehicles use it with few or no engine modifications. It also helps solve crime. Applying biotechnology to legal or criminal matters is an important part of forensic science. DNA sequencing can determine family relationships, such as who is the biological father of a child. A major application for forensic DNA is using PCR to retrieve DNA left by the perpetrators at the crime scene. The sequencing of the DNA is characterized and then compared against a database of DNA profiles of people with convictions. In the United States, a national DNA database contains a DNA profile of over 10 million people who at one point were convicted of a serious crime. As of 2013, comparisons of DNA profiles from crime scenes with the convict DNA database have produced over 200,000 matches, so hundreds of thousands of crimes have been solved with this method. DNA is also used to identify the remains of victims of mass disasters like tsunamis or plane crashes so they can be returned to their families. Over 800 victims of the 9/11 World Trade Center attack were identified entirely by DNA. This was done by sequencing DNA recovered from the remains and comparing it to DNA sequences from samples donated by family members. So, basic research is research that tries to understand how things work without the goal of whether the results will have a practical real-world application. Much basic research tries to understand the molecular basis of biology, how plant and animal cells function, and how DNA works. Basic research doesn't sound as fun as applicable research, but you can't start doing stuff to have an advantage to the society if you don't understand how it works, and that's where basic research comes in. Most basic research is done in university labs with government funding, but larger biotechnology companies often devote part of their budget to supporting basic research as well. This is because the understanding of biology that comes with basic research often leads to important methods with many applications. Today, we have reviewed 4 major methods in biotechnology, all developed since the 1950s. DNA sequencing allows us to determine the order of nucleotide bases. rDNA allows us to move genes between different organisms. SCNT lets us transplant an entire nucleus into a new cell. PCR allows us to make many copies of a specific segment of DNA. These 4 methods are used in many fields, including medicine, agriculture, environmental work, and forensic science. The foundation for most techniques in biotechnology comes from basic research, which is targeted at improving our understanding of how things work without the goal of whether or not it has practical value or not.